Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Welcome everybody to the postprandial talk of the day on virtual research environments and collaboration. Joe Futrelle from NCSA is the first talk on Medici. Joe?
Hi, I'm going to be talking about Medici, as I'm told by my Italian colleague Luigi to pronounce it, a scalable multimedia environment for research. And this represents some work to extend semantic web concepts to building a research environment for organizing multimedia. So why multimedia? Multimedia is relevant to scientific research because scientific data increasingly includes large volumes of high-resolution multimedia objects. The explosion in sensor density and data volume means that we have lots of high-resolution images, we have lots of high-resolution audio and video, and then we have complex scientific data formats and metadata standards, which themselves are sort of multimedia. If you think about an HDF file with lots of multi-dimensional arrays, those are sort of media. They're not really text, for instance. And researchers need to manage these large volumes. They need to organize and track data throughout the research process, so they need to understand where data came from and where it went. They need to share data both before and after publication. So they need to share data with their work colleagues, and then once a publication comes out, people need to be able to access the data. And they need to preserve the data for posterity. Data is being collected now, which is going to provide insights 50, 100 years from now. So we need systems that can cope with that. So multimedia. So could we just put scientific data on Flickr or YouTube? Well, those web-accessible tools are nice. They're generic. You could put all kinds of photos in Flickr. They could be photos of scientific value or not, and Flickr doesn't care, so that's good. Users really like not having to manage storage. People would rather, for instance, be able to access their photographs from everywhere rather than having to keep them all on one hard drive. Those systems show that metadata, tagging, and linking are really effective means of organizing information. People are used to organizing information in folders on a desktop operating system, but there's no such concept of folder in Flickr and YouTube, but yet you find that people have organized the information coherently, relatively speaking. But there are some things that make Flickr and YouTube poorly suited, and sites like them, poorly suited for scientific data. There's really no community ownership or control of resources in those systems. So Flickr could go away tomorrow, and my group of people sharing photos on Flickr couldn't really do much about that. There's also relatively inadequate privacy. For instance, if you wanted to maintain a collection of unpublished work and share it only among a small group of people, that's a little awkward to set up on those sites, especially YouTube. But there's a number of examples where the privacy controls aren't quite where you would need them to be for certain kinds of scientific activities where you really need to wait before you're ready to publish. There are limits on file formats, volume, throughput, and resolution in all of these online multimedia systems because they're carving out a little piece of service, quality of service for every user. And there's millions and millions of users, and we don't have that many users in science, but we have much higher data volumes. There's also not support for doing domain-specific processing. So if I routinely need to process a video in a certain way in order to get a scientific result, I can't plug that into YouTube and have YouTube do that processing for me. All it does is produce the thumbnail video that I can view. It won't let me plug in my own codes. There's also no real notion of provenance in these systems, which is very critically important for science. In a lot of these online systems, everything is just a stream of posts. So I can see how they're time sequenced, but I might not necessarily even be able to go back 10, 20, 30 days because they're just optimized for pushing the latest stuff at you. So how is Medici designed to address these problems? We've designed Medici from the ground up to be pluggable, extensible, and scalable. Pluggable meaning you can plug your own codes in. Extensible meaning that we can apply it to different domains relatively easily without having to write a new application or a bunch of new code. And scalable in the sense of scaling out. We can support multiple communities using a single code base. Communities can manage heterogeneous information without having to use more than one application. We give the communities ownership and control of resources. So we don't, for instance, position Medici as simply a cloud service where we are putting it up. We can host Medici instances for communities, but they can also download the software and run it on their own server. It's all based on open source stuff. We use a standards-based semantic content model, so all information is represented in RDF in Medici. So we don't just have a multimedia schema, for instance, that we're supporting. So if a scientist has some interesting kind of metadata they want to attach, that amounts to writing some new metadata in the system. We don't have to develop a new schema or a bunch of new interfaces. Provenance is based on the open provenance model, which is work that's come largely out of the scientific workflow community, but we think is more generally applicable than that. And so there is at least some nascent standard developing around provenance, and the one that we're supporting at the moment is the open provenance model, but if a standard comes out, we'll support that. And then we provide multiple access modalities. So if you want to use it on the desktop, we have a desktop client. If you want to use it on the web, we have a web client. If you want to write scripts against it, we have a bunch of APIs you can write scripts against. It's all open source, and it has public APIs. So if you want to extend it, there's a bunch of different ways to extend it. Some of them are really, really trivial. Some of them you have to get into the guts of the code or know Eclipse RCP, et cetera. The architecture is that we have a couple of front-end applications that provide a set of generic capabilities for organizing multimedia information and interacting with it. We have a web app. The web app has two services behind it. One of them is Tupelo, which is the semantic content middleware that takes the underlying storage resources and makes them look like RDF triples to the layers above them, so that we can elide over some of the distinctions between how those storage resources are organized and allocated. And that's important for scaling, so that we don't have to put everything in one database instance or one file system. Then there's an extraction service with extension points for different file types that is a distributed service that we can run on as many nodes as we have to, that gives us CPU cycles for performing domain-specific processing on different file types. And that has the pluggable extension points, so that if you have a code that can, for instance, extract a certain kind of metadata from a certain format, you can plug it into the extraction service, and then on ingest it gets processed. And all the results go back into Tupelo. Tupelo is a lightweight middleware fabric, so it's not a triple store. It's a broker between applications and underlying ways of storing and retrieving information. Not only does it manage metadata, but it manages binary data as binary large objects, each of which is addressed with a URI. Again, if the underlying storage system doesn't address them by URI, so you provide a mapping function. It has this standard portable model of metadata. It's linked data style RDF, so we don't require that everything has an ontology up front. We don't do a lot of validation or inference because we think those are edge cases, and the main case is just moving information around and making sure that we can link things together, even if they don't all come from the same, even if they're heterogeneous. And then Tupelo puts this context model in front of resources so that you perform generic data and metadata operations against the context, and it decides how to route them and implement them based on how it's configured. And the badges there are for some of the underlying technologies that we can use to actually do the storage and retrieval. But you can plug in other ones. It has an open API for doing that. That's the context API. The extraction service provides us with multiple extensible pre-processing pipelines, so that when a researcher uploads a file of a certain type, it goes through a processing step that can do a number of things. It can extract metadata. It can produce derived data sets with provenance, linking them back to the original data set. It's recursive, so when a derived data set is produced, it goes through the same ingest process, and if it has some extraction service plugins associated with it, that extraction will run. And we use that for a variety of things, which I'll show off later. We use it to produce the web viewable previews that get displayed in our web interface. So you can upload a very high resolution image, but you don't have to download it in order to see it. You see the web viewable preview. And we can use it for all kinds of domain-specific processing. So you can, for instance, we have an application that extracts metadata from FITS headers. We extract geolocation data from JPEGs. You can do feature detection if you were a data mining person. And we have an example where we have researchers studying 18th century, not manuscripts, but typewritten, typeset 18th century texts that have unusual typefaces, so you can't use standard OCR. So we plug non-standard OCR into the extraction service. They upload a bunch of page images, and they get the text back, and so then they can search and they can see the provenance relationship between the extracted text and the specialized OCR process and the original image. The client features are pretty straightforward. These clients are designed to look a lot like the kind of social media clients that people are familiar with out there, because they provide a number of data organization modalities that we think are generic enough that they apply to multiple domains. So you can upload and download, you can search and browse, you can tag, you can comment, you can build collections. They don't have to be hierarchical. You can put domain-specific metadata annotations on them. You can run content processing on your own desktop. It doesn't all have to be done in the extraction service. And then there's various kinds of content type-specific previewing. So if you have an image, you can zoom in on it using Microsoft Seadragon. If you have a movie, we play them with a Flash player after generating a lower resolution preview movie. If there's a 3D object, we're plugging in some HTML5 applications to allow you to rotate and view the 3D object. And we can extend this as needed for multiple scientific domains by plugging stuff in. And it also provides mapping for geolocated data, and then you can browse the provenance information. So there's an interface to the provenance information that's simple. And here is, I'm going to show you a demo movie, if I can control this while looking at it sideways, that shows us some of the features of the web client. It's built using GWT, and so it's sort of an AJAX application. And here's a whirlwind demo of some of its features. Here you see browsing multiple data sets. You can page through them. Data sets have previews of various sorts. These are image previews for the most part. There are collections that have been built, and so here I can look at a bunch of microscopy images in this collection. When I look at one of them, I get a bunch of metadata about it. I can see which collections it's in. I can add tags using a sort of AJAX-like interface, or actually a real AJAX interface. And here's the data set it was derived from. I can click on that, and I can see that a pseudo-color transformation produced that. I have a tag cloud. So here I can find a data set that's been tagged. This is a 730 megabyte high-resolution microscopy image. Here's a comment about it. Here's some metadata. Some of this metadata has been extracted. You can see some domain-specific fields that can be added. And those are, the list of domain-specific fields is stored in Tupelo. So that can be extended just by writing data to Tupelo. We don't have to write new interfaces. Seadragon provides us with the ability to zoom in. The extraction service is producing the tiled images. So this is really handy when you have extremely high resolution images. The microscopy, the researcher who has these microscopy images is going to want to get down to the level of these features. And so they haven't had to download the data. It's all, the tiles are all streamed as they're browsing. And this is Microsoft Seadragon. It's all written in JavaScript. There's no plugin required. We have full text search. So here I'm searching for movies. I found a movie, and now I can play it with this movie player. I think finally, I'm going to demonstrate geolocation. This is an image of the Morrow plots, which is an experimental field that's been planted with corn every day for over 100 years on the U of I campus. This picture was taken with an Android phone and uploaded directly using an experimental Android client to Medici, which extracted the geolocation data and wrote it out in GML-compliant metadata terms. Here on the Google map, you can see the Morrow plots. You can see the location of the sign where the picture was taken, and then you can zoom out to get more information about where this all happened. So citizen science is an obvious application for this kind of thing. People have cell phones with GPS in them, and we could start to collect and map information using this system. But it's also a system, like I just showed, that can support various kinds of other more domain-specific work, like examining these micrographs. Moving on, here is. This is where I wish I knew the key. This is the desktop client. It provides a lot of the same features as the web client. It's written in Eclipse RCP, and then you can process stuff on the client. And what's interesting about this is that it is extensible. I don't mean extensible via Eclipse RCP. What I mean is that the processing tools themselves, the code that is used, for instance, to generate this thresholding, is stored in Tupelo as a bunch of Java binaries. And the Cyber Integrator workflow engine is responsible for enacting the transformation when the user wants to run it. And so we can extend this, again, just by writing data to Tupelo. We don't have to write new code to provide a new tool to the end user. And the tools have their own interfaces, et cetera. And so we basically reused parts of the Cyber Integrator workflow engine to allow the users to run domain-specific processing on the client. All the data goes back to Tupelo, and all the provenance is captured so that when I do a transformation like this, the provenance trail shows up in the interface. So future directions on this, we're working with a lot of pilot communities. We want the users to be able to link data sets together using associative links that are from domain-specific terminology. So we're working on the interface for that. That's, we think, a very general capability that will be useful to a lot of people. We are also adding more user management of the access control system that we have. Building access control on top of triples is a new kind of thing, and we're learning how to do it. We want to provide some sorts of inference. Again, we don't think inference is the main value of the semantic web. We think it's an edge case. But for instance, we find that when users want to organize things into collections, they want to associate properties with every item in the collection. And here we can take advantage of RDF and we can infer the item level metadata from the collection level metadata. And this builds on work that's been done at the library school at the University of Illinois. We obviously want to support standards for moving data in and out of this system. We don't want this to be a closed system. We want it to be open. That's why we give everything a global ID and represent everything using standard terms. And so we want to support protocols like OAI-ORE and SWORD. Again, we need to find a community that needs us to do that, to drive this work. We also need to support compound data sets. Right now, we do focus at this sort of single file level, and we have a number of relationships that we support between the files, but in practice, scientists really want to bunch things together and call them a single object. We don't want to force scientists to put everything into a single hierarchy that can't overlap with other hierarchies, but compound objects, as they are in OAI, turn out to be really useful to scientists. And really, that amounts to making a couple of new kinds of semantic links in our system and then building some interface Chrome around that. So it's not going to be hard to support, but it's work that we have yet to do. We also want to add support for manual provenance annotation because there's provenance that happens outside of automatic processing. And if the user just knows about where the data came from and where it's going to go, they should be able to assert that, and then it should be browsable along with the machine-generated provenance. And we also want to support cloud computing in that if we have high demand, we want to be able to provision new nodes for the extraction service, for instance, where most of our CPU cycles go. By the same token, we need to be able to scale out to very, very, very large numbers of triples, and that means not all the triples are going to be on one host. Tupelo can already do queries across more than one host, but we need the provisioning logic in Medici to be able to support that. And then we also want to be able to do on-the-fly mixing of Medici instances. So if you have two instances that have similar information, we don't have a namespace problem because everything is in RDF and we have URIs for everything. So we should be able to combine them on the fly and give the user the same interface to more than one Medici instance. There are some issues that come up there, but it's something that we're tantalizingly close to being able to do. And this starts to get out of the closed system model where I have to import and export every time I want to manage something in my favorite interface. We should be able to take multiple projects and then the researchers start working together and then just combine them on the fly. And if it doesn't work, we uncombine them, we mix more things in and out of that. So this is a capability that we're very close to being able to implement. And so we hope to be able to show that in further work. Here are some acknowledgements. A large team worked on this. We have representation from image processing, from humanities computing, and from a number of projects. And that's where some of our collaborators come from. This was funded out of the Office of Naval Research and the National Archives and Records Administration. And we are grateful also to Microsoft for Seadragon, which has worked very well for us. You can see me for a live demo. We have a demo server running that you can connect to. You can get an account really easily. You just click the sign up button, and then one of us approves the account, and then you can log in and start playing. Upload your own data, mess around with it. And then you can contact us at Medici at ncsa.illinois.edu. That's it. Questions?
Any questions? Capture this for posterity, so you have to use the mic.
Art Gaylord from the Woods Hole Oceanographic Institution. What's your estimates of the scalability at this time? I mean, thousands of objects, millions of objects?
Well, it depends on whether you're talking about one. Again, because the front end is built on Tupelo, we can partition the underlying data in various ways. So at the moment, we don't have a collaborator that has more than tens of thousands of objects. We have put a Lustre file system behind this, and we were able to get up to multi-terabytes of data pretty easily. So again, because we have some flexibility about what we plug in the back end, we think we can scale up pretty well. Scaling out, we're going to have to modify a little bit how we do some of the indexing. And there are various techniques that we can use to do that. But the idea is we want to be able to scale up to very large collections. So we're talking about millions of objects at least, and certainly hundreds of terabytes. But we don't have a customer who's got that kind of volume coming at us at the moment.
I have a question.
Yeah.
So you talked about images and videos and various other things. Given that this is focused on the humanities, are there any things that are going into textual analysis and other things? And there's also I think the Caesar project at UIUC. Is there any overlap between that sort of virtual research environment for textual analysis in your work?
Well, actually, I'll correct you and say that this isn't focused on the humanities.
Okay.
And so one of our customers are a bunch of molecular biologists, like I showed you. And so we're really hoping that would support scientific data generally. But we don't have a customer who's doing NLP, for instance, or large-scale analysis. Really, the system is for organizing and managing data. It's not really, and it provides some tools for analytics, but it's not a large-scale analytic system. So there we think that a system like Caesar is probably the more appropriate choice. But then when you do the analysis and you produce a product from analysis, you still need to organize it and preserve it. And so that's more the side of the use case that we're trying to hit.
Okay, thanks. Any other questions for Joe? Okay, well, thank you very much.
Thanks.
So I'm approaching this from the blogging side, so I should be able to give a fairly good summary of the environmental data side, but I may not be able to answer some of the more difficult questions. Environmental data needs to be visualized. When you're just faced with a large number of numbers in a data set, it's kind of hard to see the features, so obviously a pictorial representation is very useful. You can spot problems in your model very, very quickly if you see there's a large black splodge in the middle of it. These data sets can be very large, so the ability to preview is a must. And just making sense of large data sets as well. You can put it into a wider context by comparing one set of data with another. And you can also communicate fairly complex concepts just by seeing it as a picture. Before we started the project, there were several visualization sort of tools. You've got for number crunching and that sort of thing, you've got MATLAB and IDL. For visualizing 3D stuff, you've got MyAvi and the Silicon Graphics stuff. There's also several web-based tools, one of which is Live Access Server. And you've got the geographic information systems. Now, these all are quite heavyweight bits of software. They tend to require a certain level of expert knowledge. And they're very, because they're all commercial, or most of them, there's no sort of interoperability between the systems. So at the University of Reading, they developed this tool called Godiva 2. It's a web-based tool for displaying oceanographic modelling data. It presents like a Bing Maps kind of interface, so you can zoom in at data, which is very simple, and most users know how to use that kind of interface, so there's no learning curve in using the system. So I've got a little screencast that sort of showing the software. Before I started, one thing you can do is, we mentioned it was 4D, so that you can actually browse the data through time and create animations. So the user can select a data set, zoom in, and we're now going to build an animation here by selecting a series of time steps. So yeah, it's very intuitive. So yeah, and now you can very quickly see the data. Now, this sort of work has always been done in isolation. You know, someone looks at their data, and in the past, the only way to share it would be to take a screen capture, maybe email a link around. So there's been no tool to sort of to handle the collaboration. So, you know, you can discuss the model with other users if you have a collaborative environment. They don't have to be necessarily in the same site, so you can, talk to people all around the world. And an important part of scientific process is the need to record it, mainly for prosperity. So in Southampton, I've developed this tool called Trove, which is basically a blogging ELN. It's open source. There's the website there. It's a web-based blogging tool specifically designed for the practicing scientist. It has been used in many cases as a collaborative tool, so less with the ELN, just more, sharing research within a group. A number of open scientists have used it to get their lab books on the web as quickly as possible. So as soon as they do their experiment, it can be grabbed by the search engines and available to anyone. It also supports the RSS feeds for syndication. And there's also the normal comment ability to allow colleagues to put their input into the discussion. Things you get with Trove is there is version control, so that the historic record, so as you see the post grow, you can actually step back in time. There's a reasonably complex metadata framework to enable the sort of classification and the sorting of all the data. And it has quite an extensive plugin-based architecture for customization, which we took, which enabled us to sort of bolt the two tools together. So the idea is that we have our Godiva sites, and there could be, it doesn't have to be any particular Godiva site. They will all talk to any blog engine. The blog engine can then build a blog post based on your bit of data that you've selected. It can then push this out as an RSS feed. Because this is all location-based data, we've also put a geodatabase behind the blog to enable geographical queries. And then with that, we can combine the RSS feed and the geodata to allow you to plot it on third-party applications. So key features. In order to do the identification service, we've used OpenID because it was a very simple sort of technology to implement, and we were dealing with more than one institution, so we couldn't use the institution's identity providers. All the integration between Godiva and the blog have been done with REST API. And we're using PostGIS as our geodatabase, still the spatial stuff behind. We managed to find some users who are willing to test the system. So this is the HiJen Project, which is a research group up at the University of Reading, and they were happy to use our tool. We very quickly gave them a prototype, and they said that the privacy controls were regarded as essential. So these weren't a bunch of open scientists. They kind of wanted their privacy. They said content is king. Well, I mean, it's a well-known phrase. They're saying that the VRE had to display the data as they wanted it. And the first system didn't actually support the animations. So the prototype had to be changed relatively quickly so they could then blog the animations. So this will, as a screencast of actually going through creating a post. So this is a standard user can create a title and some text. And so it's gone away and it's actually sending the package over to the blog server. And then you can now see that the animation can be viewed and played back from inside the blog. So once it's in the blog, so it's actually moving a bit quick, but you can search and obviously it can push out the RSS feeds. We said you've got the geo RSS, so we can take that same RSS feed and put it in a third-party application like Bing Maps, and you can easily see where the regions of interest are. And obviously, by clicking on a particular post, you can sort of take the steps back to the blog. Future features we want to implement is actually be able to put blog posts on some more of the immersive applications. We also want to actually be able to query the database with the RSS feeds to say, look, I want just the North Atlantic, so you can give me feeds there. And Godiva supports other representations of the data in terms of you can go through a depth of the data. So we would like to get the blog to start being able to display the sort of other parts of data. I would like to acknowledge the members at the University of Reading, so that's John Blower and Edit, and the HiJen team who are willing to use it, and from the University of Southampton, Jeremy Frey. Thank you.
Questions for Andrew? So I'll throw one out.
Okay.
You mentioned some of the user feedback that you got initially, and content was king, and the users only wanted to see the information that they were interested in.
Yeah.
What did that mean? What is it that you needed to change in the original version that they saw that they weren't getting?
We kind of tucked the actual data behind it away.
Okay.
And unfortunately, that screencast didn't actually have the map, but the current product actually has the full sort of image on the post.
Yep.
So it's very much bringing that to the thing that they were browsing on when they sort of scrolled down the full data.
Okay. Okay. Jeremy?
Obviously, I wasn't here quite at the beginning. I can, because I can make a fool of myself. At the moment, there's going to be one blog attached to a given Godiva server. I think that's right. But the question in the end is, do you think people will want to have separate blogs, or will there be just one community blog for everybody? Because the Godiva system itself is split over quite a few sites.
Yes. At the moment, that is true. There's only one Godiva site that supports this, and only one blog implementation that supports this. But because we're using the REST API, so it's passing a URL around, that URL can be given to any Godiva instance that's on the latest version, and that can post to any of the Lab Trove installations. So that it's not tied down to a particular Godiva site and a blog site.
And are users able to, if they've gone through the steps and created the animations, actually post those animations up into their blog as well?
Do you mean post them into like their blog spot and sort of other third party?
Right.
Yes.
Depending.
Yes. The blog will expose all that data as URL queried forms, so it can be posted anywhere.
Okay. Any other questions for Andrew? Okay, well, thank you very much. Okay, the third speaker this afternoon is Matthew Horridge from the University of Manchester. He's going to be talking about RightField, rich annotation of experimental biology through stealth using spreadsheets.
Okay, good afternoon, everybody. Welcome to this talk. As Alex said, I'm going to be talking about a tool called RightField. And I'm Matthew Horridge, and this is work that I've done with my colleagues at Manchester, Katie Wollstonecroft, Stuart Owen, and Carol Goebel.
Hey, and you stop.
And also, yeah, stop the recording. And also Olga Krebs and Wolfgang Müller from HITS Heidelberg. So I'm going to start off with a bird's eye view of what RightField is, just to say what it does and to set the scene. And then I'm actually going to explain why we developed it and go into a bit more detail of how it actually works with a demo. So what is it then? Well, if we have an ontology, and an ontology being machine processable artifacts that represents the relationships between some terms in some domain of interest. We have an ontology. We can use RightField to take a portion of the terms in the ontology, and then we can take an Excel workbook, either an existing workbook, or we can create a fresh one in RightField, and we can take the terms and embed them into the workbook. So we can mark up the workbook so that cells in the workbook, the values that they take are constrained to values of terms in the ontology. And once we've done that, we can take that and dish it out to people, and they can populate it and fill the values of these cells in the workbook. And we can then take all of these spreadsheets back. Once we've got them populated, we can extract this information and store it for reuse. In our case, we're going to store this in an RDF graph. And this is hopefully useful for querying, reuse, and repurposing of this data. So this is a very high-level overview of what RightField does and the context in which it's used. And now I'm just going to go into a bit more detail of why we actually built it. It was developed as part of a project called SysMoDB, and this is a project to do with systems biology of microorganisms. And on this project, there are approximately 300 systems biologists working, doing lab-based experiments. There's lots of sub-projects with lots of partners per sub-projects. And they need a way of publishing their data and sharing their data between them, so between the partners on each sub-project and the sub-projects overall. And there's lots of different kinds of data, and they've got lots of different formats. So they've got their own databases, they've got their own file systems, file formats, and there's a limited curation of resources. There's limited curation resources, sorry. So there's a need for collecting this data and sharing it amongst them. And the SysMoDB project was it was set up to do this and to facilitate this exchange of data between them, between these research groups. So the main goals were to describe experiments and the results of experiments. And in fact, in this area, there's fairly recently, there's been quite a lot of work done on this and specification of what are known as minimal information models. And these specify the minimum annotations and minimum metadata elements that you've got to attach to your experiments and experimental data. So they consist of things like guidelines, checklists, and these metadata elements. And they say things like what kind of experiment was performed, who performed it, what was measured, what were the experimental conditions, what methods were used, the apparatus, and so on. And there's a drive in this area towards this standardization. So in fact, some journals, I believe, won't accept submissions for publication unless the experimental results are accompanied by these minimal information model instances. But despite this drive towards standardization, there are a few tools to help scientists manage this, and RightField is hoping to fill this area. Now, one of the key things about this is that these minimal information models often use controlled vocabularies and ontologies to define these metadata elements, and this is necessary for the purposes of sharing. So in the SysMo project, we've got these things called Germs, and these are a kind of minimal information model. So Germ stands for Just Enough Results Model, and basically it's the specification of the minimal amount of information needed to find data and the minimal amount of information needed to interpret data. And different types of data require different germs. So an experiment to do with microarrays, for example, requires a different germ, maybe slightly different germ for an experiment to do with NMR proteomics. And in SysMo, germs are specified using Excel spreadsheets. So we have these germ templates, and these take the form of Excel spreadsheets. And SysMo provides a kind of starter set of germs, if you like, for common experiment types. But then all of the partners in the different projects in the SysMo project want to eventually create their own germ templates or extend existing germ templates. So we've got this starter set, but they're going to be extended eventually. And the idea here then is, as I say, to specify these germ templates using Excel spreadsheets. So in the spreadsheets, we have the fields in spreadsheets and the values of these fields, and are specified using control vocabularies. Because if you recall, we want to share these, we need some way of controlling it. And if experimental biologists are going to extend and create their own germs, we still need some control over it. So the purpose of these control vocabularies and ontologies is to define precisely what the fields are for population and what values they can take. So the goal of RightField then, as you can probably guess now, is to make it easy to create these germ templates and embed this ontological information, these control vocabulary terms, into an Excel workbook for population by biologists. And ultimately, when the biologists get these templates, they don't realize they're actually doing this kind of rich annotation. So, as I say, with RightField, we want to make it easy for them to do this. And why do we want to use spreadsheets? Well, if you recall, we eventually want to turn this into RDF, and bring all these resources together for querying, etc. But I suppose in this case, the alternative would be to use something that could build RDF graphs. So something like Protégé. And this is undesirable for several reasons. So first off, people would have to learn how to use Protégé. It's a fairly complex tool, fairly steep learning curve. It's about ontologies. Again, learning these formalisms behind ontologies, ontology languages like OWL or even RDF schema have got a fairly steep learning curve as well. The biologists don't want to keep up to date with all these standards. So tools like this aren't ideal, whereas with spreadsheets, these are everyday tools for biologists and people who work on these projects. They know what they are and feel comfortable working with them. And we found that they don't even actually want to work with fake spreadsheets. So there's tools and plugins for Protégé that produce these kind of matrix views. And they're in the spirit of working with a spreadsheet, but in general, these people who are going to be using this want to keep the spreadsheets. They like the formulae in them. they like the macros in them, the charts, etc. They would lose all that if they had these kind of spreadsheet simulation tools. And they don't even want something that's almost got the same look and feel. They like the familiarity with Excel. And we don't want to make the barrier to annotation any higher than necessary. OK, so with that in mind, I'm going to show you now how you actually embed the terms into a spreadsheet using RightField and give you a flavor of the tool. So this is the RightField tool. And what you see over here is we've loaded an Excel workbook. This is the left-hand side. And on the right-hand side here, you can see a kind of inspector where we can see any ontologies that we've got loaded. And we can see configuration information about the cells in the spreadsheet. So what we're going to do now is load an ontology from the bio portal at Stanford, in this case, the MGED ontology for microarray expression data. And we'll just load this. We get a little representation of the class hierarchy. I mean, this isn't designed for browsing ontologies. You can do that with the bio portal. But to mark up a term, then, what we actually do is we select the term in the workbook, and then we search for something in the ontology of which we want to mark up, use to mark it up. In this case, we want experiment design type. And the values for the cell in the workbook are going to come from instances of the experiment design type class. So we can choose various options here with which to generate the list of terms from. And we just go through selecting these cells. So once we've marked up a term, you see it appears in green here. And if you select it, you see the markup information in this inspector. So we go through this, and eventually we get the spreadsheet marked up. And we can save this as a workbook and then give it out to our biologists, the experimental biologists in the lab to use for describing their experiments and annotating their data. So just to make the point as well, in the demo there, we were selecting single cells, but we can actually select ranges of cells and whole columns rather easily for markup as well. So you saw that we loaded an ontology from the BioPortal repository in Stanford. And RightField supports loading ontologies in various formats, various ontology languages. These are the three main ones. So we've got RDF schema. This is a fairly lightweight ontology language with subclass relationships and instance type of relationships. The web ontology language are much more expressive there. We can load those kinds of ontologies too into RightField for term embedding. And the one we loaded in the demo was an OBO ontology, Open Biomedical Ontologies. And all of these types of ontologies can be found in the repository at Stanford. It's BioPortal repository. This can be used for searching the ontologies and browsing them as well for people to find ontologies, what they want to use in their germ templates. And there's a direct connection to this in the RightField tool. But one of the key points of this is that it contains published ontologies, well-known ontologies. And this is really necessary for us because when people create new germ templates, we need to know where to get these ontologies from. They need to be have some kind of quality about them. And also importantly is that the BioPortal contains multiple versions of ontologies. It tracks different versions. So as ontologies evolve, we can compare which ontologies we use to create a germ template and to annotate experiments with, and we can always go back to those ontologies. So this is rather important, and the BioPortal provides these resources necessary. You can also load ontologies from the local file system or directly off the web, but this is slightly more powerful. So once we've marked up our spreadsheets and saved that, how does that look in Excel? Well, here I've got a shot here of the spreadsheet that we marked up, the workbook. And what actually happens, I don't know if you can see this too well on the screen, probably not, but you get the marked up terms. They've got a slightly yellow background in here. And to the biologist, it just looks like a normal spreadsheet that they've got to fill out. They can type all the free text in, but when it comes to these fields, what they have to do is select one. So if they select a field, you get the little drop-down button, and then you get a list of terms there. And these are the terms that we extracted from the ontology in our spreadsheet. So here we're just going to select something like a data coder, and that goes in from this drop-down list. if we try to edit the actual value to something that's not in there, we get an error message saying, well, you've got to stick to values from this list. So we enforce this nice annotation. This is just vanilla Excel provided by the data functionality in Excel. So you saw this drop-down list with the terms in there, but we also embed more information into the Excel spreadsheet that enables us to do this. And we embed information in there to track the provenance of stuff. So it's really important to be able to say if the experiment design type was X, exactly what X is and which ontology it was defined in. So not only do we have the human readable names embedded in there, but for each one we've got the term identifier. This is the unique term identifier, the IRI. So we can always go back and look at an ontology that contains that, and we know we're talking about exactly the same term in a given ontology. It's not just something that happens to have the same human readable label. We also embed information about the ontology that defines the term. So we can have multiple ontologies that contain definitions for the same term. So it's important to look in the right context of where the definition is. We store that. You saw the version information in BioPortal. OWL itself has got inbuilt version mechanisms now, and so we store the versions of the ontology that was used to define the term. And finally, if it's published on the web or indeed in BioPortal, we store the physical location in the actual workbook as well, so you can always go back. So all of this is really important once we come to the next step of extracting the data. The only place where people really see this in the user interface in RightField is when the administrator loads a workbook that contains markup information in it. So if they load that, they get this message saying the workbook contains this information, and they get asked whether they want to load it, and we can track this rather easily. If they don't load it, they can still load the workbook in RightField for processing or checking. We just don't have the ontologies, but we get all the list of terms and the marked up cells with the values in them. So the technologies that we use, well, we needed it to be cross-platform, so RightField's written in Java, and it's platform independent. And the other reason for this is that a lot of the libraries for dealing with semantic web stuff, such as OWL, OBO stuff, RDF, schema, written in Java as well, including the OWL API, which we use for loading our ontologies. This supports all these different formats. And we do a bit of reasoning to get the class hierarchy and compute instance types, for example. And also, we use the Apache libraries, the POI HSF libraries, for loading and saving the workbooks and manipulating them to embed this data and reading the data stuff back from them. In terms of technologies on the Excel side of things, well, it's just vanilla Excel. There's no special add-ins or anything like that. This is just the data validation functionality. These drop-down lists are provided by Excel out of the box. And this was really important, again, for cross-platform purposes. So we didn't need any Visual Basic code or anything like that to help us here. And as far as the biologists are concerned, they're just entering their experimental data into Excel still, and they don't really see any changes. They don't have to load any special plugins or do anything any different. This is a real tool. It's available for download. It's deployed. It's being used in the Sysmo projects. And you can get it from this link here if you want to have a play with it, www.rightfield.org.uk. It's not specific to biology or microorganism biology. If you've got any sorts of ontologies, you can just use them and it should work. So just to round off then, I just want to acknowledge some of the usual suspects, and in particular Stuart Owen, Katie Wollstonecroft at Manchester. Stuart's now taken over the development of this. Carol, I've got her name spelled right there. She's led the project, keeps us off the streets, that sort of thing. Wolfgang and Olga Hitz in Heidelberg as well. They're our project collaborators on Sysmo. So that wraps it up, really. You can download the tool from there. If you want to get any more information about Sysmo, SysmoDB, there's a link there with plenty of information about the tools. Or you can always speak to myself or Carol. Okay, thanks.
Fantastic, thank you. Questions? Jeremy? Yeah.
I'm going to have to admit to being a little baffled. So, RightField, you'll have to correct me if I get any of this wrong, is a tool for generating these germ templates.
It's not specific to the germ templates. That was the use case. It's
Okay.
just a tool for adding this, embedding these ontology terms into an Excel workbook to provide functionality so that people can choose terms for the values of fields, and they don't choose incorrect terms or mistype them, things like that.
I see. So you could put a completely differently organized spreadsheet in there, and then you
Exactly.
could constrain the values in a column to
Exactly.
be from some controlled vocabulary that was associated with an ontology.
Yes.
And then when you have a spreadsheet like that, what process turns it back into RDF? Or is that just not covered in your talk?
So this isn't covered in my talk, mainly because it's not implemented yet. But it's in the plans to do this. And there's various tools that do this. I think Excel 123 is a tool for dealing with spreadsheets and RDF.
Excel wrap.
Excel wrap,
is the
sorry.
tool that we're specifically using.
Yeah.
But we've also got a license for Cambridge Semantics platform, which is a complete platform for ripping apart Excel spreadsheets and querying them in an RDF bus. That's the first step was controlling the spreadsheets, so that when you pulled out, we were able to share amongst the consortium the same data, the same templates. And the next step is how we do cross-spreadsheet query.
Yeah. I should just give my colleague as well, Simon Jupp, at the back there, a plug as well, because he's developing a tool called Populous, which is based on RightField. And what he wants to do is pretty much this, give biologists, domain experts, workbooks like this, that these people don't want to use something as complicated as a protege, and they don't want to build an ontology per se, but we want them to build an ontology without knowing it. And he's using this to mark up workbooks, and then using various patterns, and he's got some pattern language to extract an ontology out of that, and that's the same idea. So if you're interested in that, you could always talk to him as well.
Follow-up question.
Yeah.
When I look at a typical spreadsheet that's generated by a scientist, it's full of instance data. Where do new instances come from in this workflow that this whole process that you're describing?
New instances and the well, I guess the biologists people doing the experiments should generate this data as usual, but the information that's embedded into the spreadsheet would be part of the instance data then for their experiments. So,
but if I have a lot of data points, each one of them is going to have a URI in RDF.
We don't actually deal with.
This is not for data. It's for metadata that surrounds. So it's part of the MIBI standards. So it's not for the actual details of the values you took in your experiment. It's for things like being able to control which strain you're using, or the fact that you are using a strain. It's the part of the MIBI standard implementation. It's not for the actual experimental data that came off the machines or came off that you were doing in the bench.
But couldn't it be?
It could be, but that isn't what it could be. But in this particular use case, that isn't what it was for. It was for metadata.
I think that would be an interesting thing to do. I know there are some challenges with doing that with the way we've got things set up at the moment in Excel. And I'd be very interested to talk to the people who you mentioned yesterday, actually, about this. Because really what you want to do, I suppose, is type data in cells with classes, data ranges.
Exactly. Experimental ontologies.
Yeah, and I think this could be done with some hackery in Excel at the moment, but I don't think it'd be very robust. And I'm not entirely sure what it'd take to get there. But I'd be very interested to talk to those guys about that.
So, and then carrying that one step forward is whether or not these templates could then be written into programmatically, so you can actually have your workflow be outputting them into the templates in the first place. So you skip that human annotation part. So you're actually producing annotated data as part of the experiment.
Yeah.
It looks very, very nice. In my experience in trying to migrate data from the bench to these sorts of infrastructures, there's 3 classes of users. The holy grail is always the research bench scientists themselves, and I think my experience is we've had very little impact on their day-to-day practices. But there's a second layer of tools where I think this seems to fit in very well, although I want your opinion on it, which is the sort of dedicated, what I call domain scientist, who knows it's their job to migrate these things into information structures. But so far I've had to struggle with tools like Protégé, and that quickly, I put myself in that category, outstrips our ability to deal with it because our expertise is in the domain, not necessarily in the tools. How has your experience been in terms of those communities? Are you making an impact at the bench level? Do you view these things as sitting in that other level of the dedicated domain scientist?
I think Carol's better placed to answer.
Matt built a tool, but I ran the project and deployed it. So you're absolutely right. So there's in Sysmo Consulting was built for the bench biologists, the experimentalists. They do use spreadsheets actually to collect their data. So we've had some success with this because ideally you want to collect data accurately at the point of data collection, which is why the drop down and the data wasn't the problem. The data is not the problem. It's the metadata around the data that's the problem. You say, I can't remember what I'm supposed to put in for that. That's what this was to try and get around. There's a second class that has been really faster to adopt this, which is the individual projects designated curators. And they're much heavier users of this, and they're part of the team that we taught to design the templates or the spreadsheets in the first place. So although we produced these spreadsheet templates in partnership with them, they've also produced their own. So they produced their own workbooks using this. So the curators are both users of the end result and the building, the tool itself. The tool itself could never get used by an experimentalist. They'd only use the end result. You're absolutely right.
Compound question for you. The examples that you had up in the screenshots up there were against XLS files. Where are you storing the added data, the URIs, the links back to the ontologies and the terms?
The added data gets stored as very hidden worksheets in the workbook.
A bit of hackery.
So it is hackery. This is what I say. So yesterday, my kind of ears pricked up when you were saying about the new Excel file format.
Yes.
And that you can put anything in with a zip file as well. Because what we originally wanted to do was put the whole ontology into, and somehow embed the whole ontology in there.
Could do.
And this, I couldn't find a way to do this with, prior for Excel 2004, I think this was working with or something like that. And I couldn't find any way to do that with this version of Excel nicely. Because initially I thought, well, we could encode the ontology into a worksheet, but there's some size limitations there, and for really large biomedical ontologies, it wasn't possible. But I was very interested yesterday when.
So let's chat later today because I've got two things that could actually.
Okay, great.
There, so.
Yeah.
Good. Other questions? Okay, well, thank you very much.
Okay,
I think it's
thanks.
time for a break. So thank all 3 of the speakers this afternoon. It's been fun.
Thanks.